Chemistry of carbohydrates, part 1. Let us look at the specific learning objectives that we will be looking today. First, we are going to define what are carbohydrates, define sources and functions of carbohydrates, classify carbohydrates to example of each class, discuss monosaccharides giving example as energy fuel, enumerate the various types of isomerism, and enumerate the important reactions and derivatives of monosaccharides. So let us begin. So what are carbohydrates? Carbohydrates are defined as polyhydroxy, aldehydes or ketones or compounds which yield these on hydrolysis. So you can look over here, this is an example of carbohydrate. Okay, so it has a lot of hydroxy groups, so it's polyhydroxy, and also has an aldehyde group over here. So it's a polyhydroxy aldehyde, and this is an example of carbohydrate, which is glucose. Similarly over here we have a carbohydrate which is again polyhydroxy. You have a lot of hydroxy groups which are attached, and you have a functional group which is a ketone group or a keto group. So this is a polyhydroxy ketone, and the example of this type of carbohydrate is fructose. Okay, so carbohydrates are polyhydroxy aldehydes or ketones or compounds which on hydrolysis will give these. So they are also represented by a general formula which is CnH2ON, where N is the number of carbon atoms. So in this case the number of carbon atom is 6. So C6H2O6. Okay, H12O6. Let's look at the functions of carbohydrates. First is carbohydrates act as a main source of energy. Why? The example is glucose. Glucose is a carbohydrate, and it is very essential for providing energy to all the cells of our body, especially to the RBCs and brain, which rely exclusively on glucose for its energy source. Okay. The brain under conditions of starvation also does utilize ketones, but RBCs specifically utilize only glucose for its energy purposes. Okay, so this carbohydrate, the most important function of carbohydrate is it provides a source of energy for the human body. Secondly, 1 gram of carbohydrate gives approximately 4 kilocalories of energy. Okay, and this is again very, very important. So when we consume 1 gram of carbohydrates, so roughly 4 kilocalories of energy is produced. Okay, and this is equal to proteins, but it is less as compared to fats, fats give approximately 1 gram of fat will give approximately 9 kilocalories of energy, and proteins give approximately 4 kilocalories of energy, similar to carbohydrates. Next important function of carbohydrate is it acts as a storage form of energy. So glucose, if we take in glucose, and glucose gives us energy. So if glucose is not utilized, glucose is converted into glycogen. So basically glycogen is a storage form of glucose. So it's a storage form of glucose which later can provide energy. So when this glycogen, which is made up of glucose, is broken down into glucose, this glucose can either provide energy, or this glucose can also help to maintain the normal blood glucose levels in our body. Okay. The next function is excess of carbohydrates is converted to fats. Okay, so when we take in excess of carbohydrate, more energy is produced, and if the energy, the amount of energy requirement is less, okay, as compared to the amount of food which we are taking in, okay, then what happens is the energy expectation is exceeded. Okay, if we are having a sedentary lifestyle, so the energy expectation is low, but we consume more and more of carbohydrates, so the amount of energy is more as what is required. So this excess of energy, the excess of glucose, is converted to fat. The excess of carbohydrate is converted to fat because we are not utilizing that glucose. Okay, why are we not utilizing the glucose? Because our energy expectation is low. Okay, because if we have a sedentary lifestyle, so we do not need much energy, so all the excess of glucose is converted to fat. Okay, so when this happens, it leads to weight gain and obesity, and we have obesity related problems. Okay, now we have seen over here 1 gram of carbohydrate gives 4 kilocalories of energy. Okay, now similarly 1 gram of fat will give you 9 kilocalories of energy. Okay, so the similar amount of similar grams of carbohydrate and fat, okay, if you take the amount of energy which is produced from fat is twice as compared to that of carbohydrate. Okay, so the energy produced is more. However, if the energy expectation or the energy which is required by our body, our lifestyle, okay, for our lifestyle we have a sedentary lifestyle, okay, so in that case this will lead to weight gain and obesity. Okay, and this will be especially more true if we consume more fat. Okay, if we consume more fat, because fat, 1 gram of fat, 1 gram of fat will give you 9 kilocalories of energy. Then the next function is it forms carbohydrates, they form complex with proteins. Okay, for example is glycoprotein. Okay, so glycoprotein is carbohydrate, glyco is the carbohydrate part and protein is the protein. So it forms a complex of glycoprotein. So this is again used for in form of various receptors which are present in the cell membrane. It acts as hormones, enzymes, etc. Okay, so glycoprotein has numerous functions. Carbohydrates can also form complex with lipids, okay, and form glycolipids. Glycolipids again are a component of cell membrane and important component of nervous tissue. Next, carbohydrates also are important components of plants. For example is cellulose. Okay, and this cellulose is something which is present in plants and which we cannot, as human beings, we cannot digest because we do not have the enzymes to digest cellulose. So this cellulose acts as a dietary fiber which is beneficial for our health, especially in those individuals who have high cholesterol levels. Okay, so this helps in maintaining the cholesterol levels. Then also it acts as a structural component. Okay, the carbohydrates, it can act as it forms the exoskeleton in case of insects, it forms cell wall in case of microorganisms, and in humans also it forms mucopolysaccharides. Okay, so mucopolysaccharides are again important connective tissue which is present in our body. So mucopolysaccharides, they provide the ground substance which are present in connective tissue, for example in joints, skin, eyes, etc. Now, what are the diseases which are associated with carbohydrates? Okay, the first and most important disease is diabetes mellitus. Okay, so diabetes mellitus is basically when the body is not able to metabolize glucose properly. Okay, so it's a disorder which is associated with metabolism of glucose. And diabetes mellitus is very common in India itself. 77 million people are affected, and it is believed globally every 6th diabetic patient is an Indian. Okay, so diabetes mellitus is a very significant problem, significant health problem, not only for India, it is a global significant health problem. Next is galactosemia. Galactosemia is again when the body is not able to metabolize galactose. Galactose is again a carbohydrate which is present in milk. Okay, so this is especially seen in newborns because newborns are exclusively breastfed. Okay, they rely basically on mother's milk which contains galactose. So if the newborn is not able to metabolize galactose, okay, it leads to galactosemia. Then another important disorder associated with carbohydrates is lactose intolerance. Again, lactose is not metabolized in our body, and lactose again is a component of milk. So this leads to milk intolerance. Okay, this is basically seen in adults. Then the other important disorder is glycogen storage disease. Okay, so glycogen storage disease is basically a disorder in which the glycogen, which is a storage form of glucose. Okay, now we have just talked about glucose is stored in the form of glycogen. So if that glycogen is not again broken down into glucose for its utilization, it leads to glycogen storage disease. So glycogen is not broken down, so glycogen accumulates in various organs, and this leads to organ dysfunction. Because these are the few important disorders which are associated with carbohydrates. Let's look at the classification of carbohydrates. So carbohydrates are classified as monosaccharides, oligosaccharides, and polysaccharides. Okay, so what are monosaccharides? Monosaccharides are made up of single sugar units. Okay, they are the simplest form of carbohydrates. It is made up of single sugar units and it cannot be hydrolyzed further. Okay, further monosaccharides are classified by the number of carbon atoms and by the number of functional groups which they contain. Oligosaccharides are the carbohydrates which contain 2 to 10 sugar units which are joined together. Okay, monosaccharide has only one sugar in it. Oligosaccharides have 2 to 10 sugar units which are joined together. And depending upon the number of sugar units, it can be disaccharides, trisaccharides, etc. Polysaccharides are when more than 10 sugar units are joined together. So they are further classified as homopolysaccharides and heteropolysaccharides. So homopolysaccharides is in which all the sugar units are same. Heteropolysaccharide is when sugar units are different. Okay, so this is an example of polysaccharide. So let us first look at what is the classification of monosaccharide. So monosaccharide, again, it is classified on the basis of number of carbon atoms and on the basis of functional group. Okay, so for example, carbon atom, if it contains 3 carbon atoms, it is called triose. And if it contains the aldehyde group, it is called glyceraldehyde. If it contains the ketone group, it is called dihydroxyacetone. Okay. So glyceraldehyde and dihydroxyacetone are carbohydrates which contain 3 carbon atoms. Okay. So both glyceraldehyde and dihydroxyacetone are intermediates of glycolysis, okay, which leads to production of energy. Next, carbon atom number 4 is, the generic name is tetrose. The example of aldose is erythrose and the example of ketose is erythrulose. Okay. So erythrose has an aldehyde group whereas erythrulose has a ketose group. Okay. Monosaccharide having 5 carbon atoms are known as pentose. Example is xylose and ribose, and ketose, those having the keto group is xylulose and ribulose. Okay, so ribose we all know is a component of RNA. Okay, whereas and these xylulose again is an intermediate of uronic acid pathway, a carbohydrate metabolic pathway intermediate. Okay. Xylose is basically a component of glycoprotein. Then let's look at the most important classification, which has 6 number of carbon atoms monosaccharide, which is called hexose, and the examples which are seen in human beings are glucose, galactose, and mannose. Okay, so glucose is the main source of energy for our body. Galactose is present in milk. Okay, mannose is again a component of glycoproteins. Okay, and we have is the hexose, which contains keto group, is known as fructose. Okay, which is present in sucrose or table sugar, and also various fruit juices. Okay, and again, fructose is also an important component of glycolysis. And last one is, if the carbon atom is 7, it's called heptose. The example is glucoheptose, and the keto example is pseudoheptulose. Now let us look at disaccharide classification. So disaccharides are classified as the disaccharide as lactose, which is made up of glucose and galactose. Okay, it is also known as milk sugar, and this is a reducing type of sugar. Okay, why is it called reducing sugar? Because here again we have is a free functional group. That means a free aldehyde group or a free keto group. So those sugars which have a free aldehyde or a free keto group are known as reducing sugars. Okay, monosaccharides, because they are the simplest form. Okay, they are the simplest of sugar. They have only one sugar unit, so the functional group by default is free. They're not binding with anything. Here, lactose consists of 2 sugar units. Okay, so the functional groups come into interaction. Okay, however, the functional group here is free. One of the functional group is free, so that is why it is known as a reducing type of disaccharide. Maltose is made up of 2 glucose units. Again, maltose is a product of starch hydrolysis. Maltose also forms osazone, that we will see. And maltose also is a reducing sugar. That means it also has a free functional group. Isomaltose also is made up of glucose and glucose. Okay, this is also a product of starch hydrolysis, and this is also a reducing sugar. Sucrose, on the other hand, is made up of glucose and fructose. So this is an example of table sugar, which is sucrose. It is also seen in sugarcane, and this is an example of non-reducing disaccharide. Okay, why is it called non-reducing? Because the functional group which is present in glucose and the functional group which is present in fructose. So the functional group here is aldehyde and the functional group here is ketone. So the aldehyde and ketone, they form a bond. So both the functional groups are utilized. So we do not have a free functional group. So this results in the disaccharide becoming non-reducing. Okay, so it becomes non-reducing, so it does not undergo chemical reactions, the reducing chemical reactions. Now let us look at polysaccharide classification. So polysaccharide, as we have discussed, it consists of 10 or more sugar units which are joined together. Now if the sugar units are same, it is called homopolysaccharides. If the sugar units are different, it is called heteropolysaccharides. Okay. So the examples of homopolysaccharides are starch, which is made up of glucose units, glycogen, which is again made up of glucose units. So starch is a storage form of carbohydrate in plants. Okay. Glycogen is a storage form of carbohydrate or storage form of glucose in human beings. Okay, so we have glycogen as a storage form of glucose, and as the need required, glycogen is again broken down into glucose, and this glucose provides energy or it maintains the blood glucose levels. Similarly, we have a cellulose, which is again seen in plants, and it is made up of glucose. So cellulose, we human beings cannot digest because we do not have the enzymes. Cellulose can be digested by animals. Okay, so in us, it acts as a dietary fiber. Okay, and we have benefits of dietary fibers that we will see later. Inulin, again, it is made up of fructose. Okay. Dextran, it is made up of glucose. Now, dextran is again a highly branched molecule, okay, which is made up of glucose, and dextran is also used as an IV fluid. Okay, dextran is also used as an intravenous fluid, which is given to patients, in those patients who are in hypovolemic shock, patients whose blood pressure is low, okay, because of, let's say, diarrhea, because of burns. So in them, if you want to increase the blood pressure, we give them dextran. Okay, why? Because this dextran is highly branched, it is made up of glucose, it is highly branched, so it is present, it stays inside the blood vessel, it does not go outside. So this helps in increasing the blood pressure. Okay. Next we have is chitin, okay, which is again present in, made up of N-acetyl glucosamine. Okay, so this is again, it is present in the exoskeleton of some insects. If you look at heteropolysaccharides made up of different sugar. For example, the simplest is agar, which is made up of glucose and galactose. Two different sugars are present. Next we have is hyaluronic acid, heparin, chondroitin sulfate, keratin sulfate, and dermatan sulfate. All these are mucopolysaccharides. So these mucopolysaccharides, they form ground substance in our body, okay, in the connective tissue, and they help in providing mobility to joints. Okay, they act as anticoagulants. Okay, so for example, hyaluronic acid, it's an example of mucopolysaccharide, which is made up of glucuronic acid and glucosamine. Okay, so this is basically a repeating unit of a uronic acid and glucosamine. Okay, similarly, this hyaluronic acid is present in the joint, it's present in the synovial fluid, which helps in the mobility of joints. Heparin is made up of iduronic acid and glucosamine. Okay, heparin is an anticoagulant. Similarly, the next mucopolysaccharide is chondroitin sulfate, okay, which is made up of glucuronic acid and galactosamine, okay, which is again present in cartilage. Keratin sulfate, which is present in cornea, is made up of galactose and glucosamine. And the last one is dermatan sulfate, which is present in skin. It is made up of iduronic acid and galactosamine. Okay, so polysaccharides are, see, the heteropolysaccharides are made up of different sugar units. Example is agar, which is the simplest heteropolysaccharide, and these are the mucopolysaccharides. These are again very important in connective tissue. Let's look at what are isomers of carbohydrates. Okay, so what are isomers? Isomers are basically compounds which have the same structural formula but they have different spatial configuration. Okay, for example, if you look at glyceraldehyde, the structural formula is C3H6O3. But this glyceraldehyde can be written like this. Its spatial arrangement, the configuration of these molecules can be arranged like this as well as like this. Okay, so if you see the difference, there is difference in the orientation of hydroxy group on the second carbon atom on one side, and here this is on the right side. Here the hydroxyl group is present on the left side of the carbon atom. So this is a different in the spatial arrangement of glyceraldehyde. Okay, so this is known as isomerism. Okay, so compounds which have the same structural formula but different in spatial configuration. Now this stereoisomerism occurs because of the presence of a chiral carbon. Now what is a chiral carbon? If you look at this carbon over here, all the four valencies of carbon are different. Okay, one has this H over here, over here it has OH. Okay, you have this CHO group over here, and here you have this CH2. So all the four valencies of carbon is different. So this becomes a chiral carbon. So whenever there is a chiral carbon, this will lead to formation of stereoisomers or isomers. Now how many isomers will be formed? That depends upon the number of the formula 2n, where n is the number of chiral carbon. So in this case, the number of chiral carbon in glyceraldehyde is only 1. This is not a chiral carbon and this is not a chiral carbon. So the only one chiral carbon is there. So only two stereoisomers will be seen. So glyceraldehyde has two stereoisomers. Okay. So now let's look at isomerism of glucose. Now glucose has 4 chiral carbons. Okay, 1, 2, 3, 4. So these are the 4 chiral carbons which are present in glucose. That means these are the carbons which have all the 4 valencies which are different, all the 4 attachments which are different. So the number of possible isomers of glucose are 2 to the power 4, which is 16. So 16 possible isomers of glucose are seen. Okay. Now the isomers which with respect to the 5th chiral carbon. Okay, so this is the carbon atom number 1, which is attached to the functional group. 2, 3, 4, 5th and 6th. Okay, so the chiral carbon which is the 5th chiral carbon or the penultimate chiral carbon. Okay, this second last one, not the last one, the second last one, the penultimate chiral carbon. So this is also known as the reference chiral carbon. Okay, so when you have isomerism with respect to this, okay, we have two types of isomers. Okay, we have is D and L isomers. Okay, which are known as mirror image. Okay, so just remember, the isomerism with respect to the penultimate chiral carbon produces mirror images, okay, which are known as D and L isomers. Okay. Isomers with respect to all other chiral carbons, okay, over here C2, C3, C4 will form diastereomers, okay, not the penultimate chiral carbon. Okay, leaving this isomers because of all the other chiral carbons will form diastereoisomers. Okay, and isomers with respect to a single chiral carbon, let's say C2, single chiral carbon, okay, excluding the penultimate chiral carbon, is called epimers. Okay, so basically epimers are a subset of diastereoisomers. Okay, so what you need to remember is glucose has 4 chiral carbons. Okay, the chiral carbon, the isomerism with respect to the chiral carbon which is the penultimate chiral carbon will form mirror images of glucose. Okay, all the other chiral carbons will form isomers which are called diastereoisomers. Okay, out of these diastereoisomers, if you see the isomerism with respect to any one of the chiral carbons, they form epimers. Okay, so we have three terms. Okay, one is the mirror image, second is diastereoisomers, and the third is epimers. Now, because of the chiral carbon, okay, irrespective whether it is epimer, whether it is the mirror image or diastereoisomers, because of the presence of chiral carbon, okay, the compound can rotate the plane of polarized light to the right or to the left. Okay, if you have a glucose solution and we pass a plane of polarized light, if you basically pass a polarized light, so the glucose solution will either shift or change the or rotate the polarized light plane either towards the right side. If it does that, it is called dextro or plus or small d. Okay, or it can rotate the plane of polarized light to the left. It is called levo or minus or small l. Okay, so this is also a property of chiral carbon, which is known as optical activity. So all chiral carbons have optical activity, okay, irrespective of the position. So this optical activity is what? The optical activity is the ability to rotate the plane of polarized light. If it rotates the plane of polarized light towards the right side, it is called dextro or plus. If it rotates the plane of polarized light towards the left side, it is called levo or minus. Okay, so let us look at the glucose isomers which are of significance. Okay, so if you look over here, this is glucose, which is D-glucose and L-glucose. Okay, so here if you look at the isomerism, it is with respect to the fifth carbon atom, which is the penultimate carbon atom. Okay, so one carbon atom number 2, carbon atom number 3, carbon atom number 4 and carbon atom number 5. All four are chiral carbon atom. But if you look at the isomers because of the fifth chiral carbon, okay, so it will have orientation of hydroxy group on the right side and the other will have orientation of hydroxy group on the left side. So if it is on the right side, it is called D-glucose. If it is on the left side, it's called L-glucose. And this forms a mirror image. Okay, so when we have isomerism or isomers developed because of orientation around this penultimate carbon atom, it leads to formation of mirror images. Okay, and it's known as D and L isomers. So this is a D isomer of glucose and L isomers of glucose. Okay, you should know that our body can metabolize only D glucose and not L glucose. So that is why this is very, very important. Okay. Next, if you look at epimers. Okay, so if you look at glucose over here and let's look at mannose over here. So if you look at the structure of glucose, okay, see the hydroxyl group of glucose and the second carbon atom is on the right side, and in mannose the hydroxy group is on the left side. Rest everything is same. Okay, rest the structural orientation of all the other components are same. So glucose and mannose differ in spatial configuration with respect to only second carbon atom, second chiral carbon atom. Okay, so this is an example of epimers. What are epimers? Epimers are again spatial isomers which develop because of difference in orientation of one carbon atom. Okay, excluding the fifth carbon atom. So here it is the second carbon atom. So glucose and mannose are epimers of, so mannose is basically an epimer of glucose at the second carbon position. Now if you look at galactose, glucose and galactose differ at fourth carbon over here. Okay, so let's look at the second carbon. Second carbon, the OH is on the right side, here also it's on the right side. Third carbon, it's on the left side, here also it is on the left side. Fourth carbon in glucose, it is on the right side, and in galactose it is on the left side. Okay, rest everything is same. So glucose and galactose, they differ with respect to the hydroxyl group or present on the chiral carbon atom, which is number 4. Okay, so glucose and galactose are epimers with respect to the 4th chiral carbon atom. Okay, so these are the 3 epimers which are seen in human body. Okay, so epimers which are seen in human body are glucose, galactose and mannose. Okay, there are other carbon atoms also. Okay, 2, 3, 4. So these 3 chiral carbon atoms also produced isomers which are called distereoisomers. Okay, but they are not present in the human body. So out of those distereoisomers, these 3 are important, and these 3 epimers are important. Okay. Let's look at the reactions and derivatives of monosaccharides. So what makes monosaccharides very reactive? It is because of the presence of a highly reactive functional group, which is aldehyde or keto. Now we have aldehyde group and we have keto group. These are reactive groups and they are not bound, they are free. Okay, they do not go into they're not binding with any other molecules, they are free. So this makes them very, very reactive. And this so the reactions are based on 2 types of property. Okay, test based on reducing property of monosaccharides and test based on furfural formation. So first let's look at the test based on reducing property of monosaccharides. Now this reducing property of monosaccharide is because of the formation of an intermediate called imidazole. This imidazole is an intermediate which is formed from monosaccharides, and it is a highly reactive intermediate, and it is formed in alkaline medium. Okay, so how it is formed? If you look over here is glucose. Glucose, we have an aldehyde group, and fructose, it has a keto group. Okay, now these form imidazole. Okay, so the imidazole is, okay, this is an intermediate which is formed. This is imidazole. The double bond shifts between the first and the second carbon atom. So this is a highly reactive compound which is called imidazole. Okay, now this imidazole forms the basis of various tests. The most important is Benedict's test, principle of Benedict's test, and also Barford's and Felling's. Now what happens in Benedict's test is, now we look at monosaccharides, sugar, for example, let's say glucose. Glucose in alkaline medium forms is imidazole, which is a highly reactive intermediate. Now this acts as a reducing agent, which brings about reduction. Now if you take let's say in Benedict's test, we have is Benedict's reagent, which contains copper ions. So these copper ions are reduced to from cupric to cuprous ions. Okay, so how this reduction is brought about? It is reduction is brought about by imidazole, which acts as a strong reducing agent, and it itself undergoes oxidation. Okay, so once you get the cupric ions are converted to cuprous ions, the cuprous ion reacts with water to form is cuprous oxide. Okay, so the amount of cuprous oxide, which is red in color, the amount of cuprous oxide which is produced is directly proportional to the amount of sugar which is converted to imidazole. Okay, so if the sugar content is high, the amount of red color produced will be, or the red precipitate produced will be more. Okay, so this also gives us an indication as to how much of reducing sugar is present. Okay, so reducing sugar forms imidazole. Okay, so this is the principle of Benedict's test. Similarly, we have other tests which is based on the reducing property, which is Barford's and Felling's. Now let's look at tests which are based on furfural formation. The furfural formation, again monosaccharides, they undergo dehydration, loss of water molecules in the presence of acid to form as furfural. Furfural again is an intermediate which is reactive, and furfural reacts with phenolic compounds to give you a colored solution. Okay, now this is the basis of Osa-zone test. Okay, so Osa-zone test, again the monosaccharides undergo dehydration in the presence of acid to form as furfural. So these furfurals, they react with phenolic compounds to give you is a colored solution or specific crystal formation. For example, glucose forms as needle shaped or broom shaped crystals, which is known as glucosone. Lactose forms a hedgehog shaped pattern, which is known as lactosazone. And maltose, again, it forms a petal shaped crystals of maltosazone. Okay, so these are all formed because of formation of intermediate called furfural, okay, which occurs because of dehydration of monosaccharides, okay, and disaccharides also. So to summarize, we talked about carbohydrates. What are carbohydrates? Carbohydrates are basically polyhydroxyaldehydes or ketones or compounds which give these on hydrolysis. Okay, we talked about functions of carbohydrates, how it is the main source of energy. Okay, the storage form of carbohydrates, or storage form of glucose, which is glycogen. How 1 gram of glucose gives 4 kilocalories of energy. Okay, then we talked about is classification of carbohydrates as monosaccharides, disaccharides, and polysaccharides. The monosaccharides have only one sugar units. Disaccharides or oligosaccharides have 2 to 10 sugar units which are joined together. Okay, so if 2 sugar units are joined together, it's called disaccharides. If 3 sugar units are joined together, it is called trisaccharides. And we have as polysaccharides. So polysaccharides is when 10 or more sugar units are joined together. Okay. So again, the monosaccharides are further classified based on the number of carbon atoms as 3, 4, 5, 6, 7, and each containing the functional group, whether it is aldehyde or ketone. Okay. For example, if it has 3 carbon atom, it's a triose. Under aldehyde group, it is glyceraldehyde, and under keto group, it is a dihydroxyacetone. Okay. Similarly, example of hexose containing 6 carbon atom. Hexose containing aldehyde group is glucose, whereas hexose containing keto group is fructose. Okay, then we talked about is isomerism. So what is isomerism? Is basically difference in compounds having the same structural formula but different spatial configuration of its atoms. Okay, so we have types of isomerism of glucose which we have seen. Okay, we have mirror images which occurs because of penultimate chiral carbon. Then we have is diastereoisomers, which occurs because of chiral carbons excluding the penultimate chiral carbon. And we have is epimers, which are again isomers with respect to a specific chiral carbon. Okay. And lastly, we talked about reactions of carbohydrates, reactions based on reducing property and reactions based on formation of furfural. Okay, so the reactions based on reducing property is because of the formation of an intermediate called enediol, and reactions of furfural is because of formation of furfural by dehydration of carbohydrates. Okay, so thank you very much. Okay, I'll see you in the second part of carbohydrate when we discuss more of chemistry of carbohydrates. Okay, so thank you.